Hi, welcome to the Bioinformatics chat. Thanks to everyone who took the listener survey. I heard a lot of very kind words and also many great suggestions. It really means a lot to me. And if you haven't taken the survey yet, please go to bioinformatics.chat/survey and answer a few questions, and I would appreciate that. Looking at the survey results gave me this idea. So unsurprisingly, many of you are scientists, researchers, and it might have happened to you that you thought, hey, my latest preprint or paper or software should be featured on the podcast. And the thing is, first, I may not be aware of your preprint or paper or software, and even if I am, it's just not possible to interview everyone. But I had this idea to host an occasional open mic episode, and this is where you would submit your audio clips. So you would record an audio clip, maybe 5 or 10 minutes long, about your research or whatever you want to share with the audience of this podcast, and send it to me. And once I have a few of these clips, I would paste them together and release this as an extra episode of the podcast. So if you want to give this a try, please go to bioinformatics.chat/openmic, OPENMIC, and I'm looking forward to hearing from you. Today I'm speaking to Amatur Rahman, who is a PhD student at Penn State. Hi, Amatur.
Hi.
And Karel Břinda, who's a postdoc at Harvard Medical School. Hi, Karel.
Hello.
So normally when I have multiple guests, they are co-authors on the same paper or they're colleagues. Today is a very interesting situation where Karel and Amatur are from different universities and they didn't co-author a paper. But what they did, in fact, was they published in January, within like several days. Each of them is a first author on the preprint, on 2 preprints that introduce pretty much the same concept from somewhat different angles. And I thought this was a very interesting situation, and I invited them both to discuss this new concept, which is called Simplitigs, or spectrum preserving string sets. So in different papers, it's called differently. But that's what we'll be talking about today. And the idea of this concept of Simplitigs is to efficiently store k-mers, a large set of k-mers. And I guess we'll start by recalling what k-mers are and why do we care about them? Why do we need to store large sets of them?
So k-mers are substrings of a fixed length of strings in a given genomic data set. And they are super important in computational biology because they are used for many different tasks. On one hand, they can be, for instance, used for like genome assembly, for instance, by programs like SPADE. On the other hand, they can be used also for rapid estimates of distances between genomic data sets. And this is very often used, for instance, in metagenomic classification. K-mers are also very often used in like variant calling in programs like Kevlar, or in database search in programs like Bixi, for instance, or in infectious disease diagnostics, like, for instance, RACE. This leads us to the question, how to represent k-mers efficiently. So what is our problem, typical, or let's say typical setting? So we have a big genomic data set, which can be, for instance, a set of reads from a sequencing experiment, or a genome assembly, or a set of genome assemblies, or reads from a metagenomic experiment. And we are interested in the question how to store the k-mers, and at the same time, how to ask, for instance, which k-mers are in there. So, for instance, we have one specific k-mer, and we are asking, is this k-mer inside, yes or no?
So the core idea here is that these are not random sets of k-mers, random sets of these fixed length strings, but these are all substrings of the fixed length k, all substrings of some larger string, right? And therefore, there's a lot of redundancy there, because if you consider two consecutive k-mers, the second k-mer only has like one extra letter compared to the first one, as it's one extra sort of unit of information. In the case of the DNA, it's extra 2 bits of information. But if you store the whole k-mer, you're storing 2 times k bits of information. And so we want to avoid that redundancy, right? So how would you approach the problem of storing these sets of k-mers efficiently?
So the first important thing to realize is how the sequencing process works. So we have some linear molecules under sequencing. It can be DNA, it can be RNA. And from the sequencing machine, we are getting a set of reads, where the reads are substrings of the molecules, basically for instance, of the genomes. And sometimes they can contain some sequencing errors. The nature of the process basically tells us that most of the k-mers will belong to certain very long strings. Plus, there might be some k-mers which correspond to the sequencing errors. And exactly as you mentioned, many of the k-mers will be very largely overlapping. So very often we will see that 2 k-mers have overlap k-1, and we can really make use of this redundancy. So we were thinking in our work, when we work on metagenomic classification, about how to store large k-mer sets in a very efficient way. And we started to play with basically leveraging the overlaps. So going basically from the full set of all k-mers, which can be either in some assembly or in some set of reads. And we started to merge the k-mers one with each other whenever there was k-1 long overlap. And we found out that this is a very efficient representation. So this is how our research started.
And that is what you call Simplitigs, right?
Yes, this is what we nowadays call Simplitigs.
And so there are sort of 2 different ways to look at Simplitigs. So far, we'll introduce more different ways later. But so far, on the one hand, this is a natural operation to perform if you want to assemble the original linear molecule. In that case, if you're actually interested in what the original molecule was, you're concerned whether you're reconstructing the right linear structure, because there can be different linear structures that give rise to the same sets of k-mers. But what you're saying here is that if we don't care whether that was the right, that was the true original linear structure, this is just an efficient way to store k-mers, because we essentially assemble them into a long linear sequence, and then if we want to get the original k-mers, we split it back into different k-mers, right?
Yes, that's completely true. So when we are just interested in the presence or absence of k-mers, the strings which we are building don't have to correspond to the original molecules under sequencing. It's rather a mean, basically, for compression for us. And it's very useful to introduce the concept of De Bruijn graphs to think about these things. So De Bruijn graph, in the node-centric definition, is a graph where the k-mers are the vertices of the graph. Plus, there is some additional information about the overlaps in the edges. So one k-mer is connected with another k-mer if and only if they share k-1 long overlap. And these k-mers versus De Bruijn graphs, this pair basically gives us very nice duality. So on one hand, we can think about it just as about a k-mer set. On the other hand, we can go from the k-mer set to the De Bruijn graph if we consider also the overlap information. Then we can think about the Simplitigs in terms of paths in this De Bruijn graph. In the assembly applications, people typically look for paths which are somehow uniquely determined, or where they can be sure that they correspond to the original molecules. Whereas, since we are interested in the compression, we are basically trying to find arbitrary paths long enough so that they can capture as many k-mers at the same time. And we basically want to minimize the number of such paths.
Yeah, and to make this clear, the reason you want to minimize the number of this path, and I guess we'll return to this a bit later, but the fewer paths you have, the more sharing there is among the k-mers, right? And so the fewer bytes you need to store. So, what one might think that what we really care is about the storage requirements, so how many bytes we need to spend to store this information. But in fact, I think there's a simple linear relationship between the number of paths and the number of bytes. Is this right?
Yes, it is right. In the best imaginary case, we would be able to basically come up with one path where every node would be visited exactly once. But for most of genomic datasets, this is not possible. But it gives us an interesting lower bound, so we always know that we cannot be better than such an imaginary path. Whenever it happens that we basically come to the end and cannot extend anymore and start with a new path, it brings us some additional overhead, let's say, into our data. Because we have to basically end the path, start once again, and we will use k minus 1 bytes in addition, or characters, let's say.
And so you arrived at this problem via metagenomics, right? So you were considering what's the most efficient way to store the large metagenomic data sets. And Amatur, you arrived sort of at the same problem, but from a different perspective. So can you describe how you became interested in this problem?
Yeah, so for that, I think I need to explain Unitics first. So I was at first looking at Unitic-based tools. So we've already discussed what the Bruijn graphs is, so that makes it easier for me to explain Unitics. So this is if you keep merging k-mers along non-branching paths as far as you can, then you get Unitic. So for example, in the Bruijn graphs, two consecutive k-mers have overlap, so you can merge them to compact the number of characters. So if you keep doing that until you hit a node which has multiple outgoing path, so then because it adds an ambiguity, we stop there. So that's how you get a Unitic. So if you find all possible Unitics in the Bruijn graph, then that gives you very that's a very interesting thing. So you get a compacted De Bruijn graph this way. So the thing is, Unitic is basically used for assembly purpose. I mean, the main reason Unitics came, the Unitic-based tools appeared, was because it helps you to speed up the assembly process. But our goal is not the assembly here. We want to efficiently store the k-mers. So we noticed that Unitics are very useful for storing k-mers because it has some special characteristics. They contain the same k-mers as the original k-mer set in the De Bruijn graph and only contain them once. So we noticed this property and realized that this is necessary for losslessly storing set of k-mers. And we gave this property the name spectrum preserving string set.
Can you define what spectrum means in this context?
So the spectrum would be if you consider a set of Unitics and the set of k-mers of the original De Bruijn graph, you'll see that you can find the same k-mers in both of these sets, and you'll find each k-mer appear exactly once in both of these sets. So that's the spectrum.
Right. So the spectrum is like the set of all k-mers, right?
Yeah, the spectrum is the property of the k-mers appearing once in both of these sets. That's the spectrum. And we are preserving the spectrum in both of these sets. That's why we call them spectrum preserving string sets. Both of these sets, I mean the original claimer set and the set of unitics, both of them are they are equivalent in terms of their spectrum. So that's the property that we notice that actually helps us to losslessly represent a set of k-mers. And then we realize that the unitics are great in this way, but you can definitely do better than that. If you reduce the number of path in the De Bruijn graph, then you get better results, like you save more characters. So that's why we're interested in a spectrum preserving string set which has like less number of strings. You can do better than unitics in that aspect. For example, in unitics, whenever we see a k-mer which has like multiple outgoing path, we stop there, we stop merging there. But what if we didn't stop there? What if we kept following the branching path? Like let's say k-mer has 3 outgoing path, and then we arbitrarily choose one of them and we keep extending through towards that path. So we realized, like we found out that if we like keep on doing this, if we keep on merging in this way, we get a better spectrum preserving string set with less number of strings and less number of characters. So this serves our purpose of lossless storage of k-mers.
Karel mentioned that there is a natural lower bound on the number of simplitigs, which is 1, and that sort of gives rise to the natural lower bound on the storage requirement of a k-mer set, right? But in your preprint, you improved on that lower bound. So can you talk about that? And maybe start with like why do we need a lower bound in the first place, or a good lower bound?
So our method of extending the k-mers as far as we can, it's like I said, it was arbitrarily chosen. Like when I had multiple paths, I picked any one of them. So it was not an exact method. It's a greedy method of forming spectrum preserving string set. So it's not optimal, we know, but we wanted to see, like, even if it's not optimal, I mean, how worse is it from the optimal result? So that's why we were interested in a lower bound. So at first we started with a very naive or simple lower bound, which so the simplest possible lower bound could be the number of k-mers. So you at least need to have at least that many strings, right? So if you have like 10 k-mers, you need at least 10 strings. So that's one of the bound. But we want.
But that's not a lower bound, is it? It's more like an upper bound.
Sorry, that's the upper bound. Yeah, that's like an upper bound, and the lower bound would be the one. Like you cannot do better than one string. So, but that would also happen when we had one path going through all the k-mers. So then we can merge all of the k-mers together into one string. So that would be the best possible result. So that's the lower bound. So 1, and the upper bound would be the number of k-mers. So but 1 doesn't give you actually, I mean, doesn't help you to compare your algorithm. Yeah, so that's why we wanted to improve on the lower bound. For that, we looked at the topology of the De Bruijn graph. So in every data set, the topology of the De Bruijn graph is different. It's also different for different k. So we made a method to calculate a lower bound for every different De Bruijn graphs. So, for example, De Bruijn graphs are what? They are sets of k-mers. The nodes are k-mers, and we have edges if those k-mers have overlap between them. So, what if in our De Bruijn graph there is no edge? What if we have some k-mers which are isolated? They have no outgoing edge or no incoming edge. So in that case, that particular k-mer has to be a single string in our set. So that's one way we improve on the lower bound. Another way you can improve the lower bound is you notice that there are some nodes which have no incoming edge. So if a node has no incoming edge, but only outgoing edge, so you know that your string has to start from that k-mar or that node. So this also tells you that you have to add this to the number of strings that you are calculating. The same goes for nodes with no outgoing edge. So these are the observations that led to a little bit more complicated models of calculating lower bound that helped us to compare our results. And we found that like if we run our algorithm in sequencing these data sets with for K31, we found that our gap with this lower bound was only 2 to 3%. So this is very interesting because the problem is potentially NP hard, like the problem that we're dealing with, the problem of finding the optimal spectrum preserving string set. But for such a problem, we found a greedy heuristic that only has 2 to 3% gap. So that's why the lower bound was important for us.
This is a potentially an NP hard problem, right? So you don't have a proof, but I think...
So we're working on it right now, but in general, like for general graphs, this is NP hard problem, but we don't know because this is a special type of graph, De Bruijn graphs. It's not proven yet, but we're working on it right now to show if it's NP hard or not. That's why I say potentially NP hard.
Right, and can you also clarify for the listeners who don't know what does NP hard mean?
So this is like this is computationally very hard to solve exactly. I don't want to give a technical explanation here. So it's going to take a very long time if you want to for larger inputs to compute the spectrum preserving string sets. So that's what I mean by NP hard here.
All right. So I guess you both made your independent implementations of the algorithm to compute the Simplitigs or spectrum preserving string sets. So Karel, can you talk about your implementation?
Our implementation is very simple. So at the beginning, we load all of the k-mers into memory, and then we repeat the following process. We take a random k-mer, and then we start to extend it to one direction, as long as it is possible. So we find a k-mer which has a k-1 long overlap, and basically merge it with our current string, with our current Simplitig, which we are right now constructing. And whenever we add a new k-mer like that, we remove it from the set. And we continue as long as this is possible, and when it's not possible, which means that there is no k-mer which would have such an overlap, we look to the left side of the Simplitig and extend as long as possible to the left side. When there is no longer possible to extend to the left side, we print a new Simplitig and we start the process again, and we repeat this until we get all the Simplitigs. So it is a very simple greedy heuristic.
Right, and just to remind our listeners, this is all happening in the context of a De Bruijn graph. So you already have this sort of relationship ready, right? You don't have to every time to search all the k-mers for overlaps, because that's the point of a De Bruijn graph. It contains the same information, but it provides this extra structure that you can traverse when building the Simplitigs.
In fact, we store in memory just the k-mers as a set without the information about the overlaps. But we don't need that actually, because for each of the k-mers, we basically can remove the first character and then just test whether the string plus A, plus C, plus G, plus T is present there or not. So in fact, on the technical level, we are working just with a k-mer set, but on the philosophical level, it corresponds to basically looking for paths in the De Bruijn graph.
So you exploit the fact that there are just 4 letters in the DNA alphabet,
exactly.
Right? Because if the alphabet was larger, that would be less efficient.
Yes, that's right.
Anything else you wanted to say about your implementation?
I would say that one of the big advantages of this approach is that it can be super easily implemented basically anywhere. You can write this code in Python, for instance, on just several lines. So from this reason, we believe that this heuristic is very easily pluggable into basically any software and can be used for like serialization of K-mer sets and the Bruijn graphs.
And Amatur, what about your implementation? Did you do it similarly, or are there any interesting differences?
It's almost similar, but our starting point was like a compacted De Bruijn graph as opposed to a De Bruijn graph. So a compacted De Bruijn graph is composed of like where the nodes are unitics as opposed to being K-mers. So I think that's pretty much the basic difference. We start with a compacted De Bruijn graph, and then we pick a node which is a unitic, and we try to extend it in both directions, and we keep extending as long as we can, and that way I get one string, and then we randomly pick another unitic and repeat the process. So I mean, both processes are equivalent. It's just if you skip the computation of compacted De Bruijn graph, and then you get the method that Karel described.
And so your method allows to store these large K-mer sets very efficiently, right, by merging them into Simplitigs. And so if you want to if you want to make use of that information, you could always uncompress, you could extract all the K-mers from the Simplitigs. But that may be not the most efficient way, right? So ideally, we would like to query this compact representation without extracting it. You both sort of investigated how to efficiently query this compact representation. And I think, correct me if I'm wrong, but you both converged on the FM index. So Karel, maybe you could talk about your approach, and maybe let's start with introducing what an FM index is.
The FM index is a compact data structure used for full-text search. So it allows to take basically any string over a given alphabet and build an index on top of it and very efficiently query it for substrings, which is very good for us because in our case, our string are basically merged Simplitigs, and then we are interested in which K-mers are present there or not. So this is our query. And the very important thing here is that FM indexes have been very largely used in the context of read mapping, and now there are many very efficient implementations, for instance BWA or Bowtie and other read mappers. So we can basically combine the Simplitic representation with any of these read mappers, more specifically with their indexes, and we can use them for querying the K-mers.
And how do you apply that concept to the compact representation?
So it's applied in the following way. So we basically take a genomic data set, we calculate the Simplitigs, we store the Simplitigs in one FASTA file, then we can build an FM index over this FASTA file, and then we can query the obtained FM index using standard Burrows-Wheeler transform search. Maybe this is an important thing to mention, what FM index actually is. It is basically the Burrows-Wheeler transform of the string plus some additional tables.
Right, so does it matter for your implementation then that this is like an actual FM index, or would it similarly work with any other way of indexing like a genome?
It could work with any full-text indexes. So it could be combined also, for instance, with a suffix array or with a suffix tree. The reason why we are using FM indexes is that they, in practice, provide the best performance.
And Amatur, you also investigated the use of an FM index applied to the spectrum preserving string sets. So how did you approach that?
It's the same method, like you first build a spectrum preserving string sets from a sequencing data set, and then it's stored in a FASTA file, and on top of that FASTA file you run an FM index builder tool that gives you a structure from which you can do query of k-mers very fast. So it's like it takes less than a second for querying each k-mers. So that's so some we can note something here, like it's not only FM index, like you can use any other index, like you can use hash based index here on top of the text, but like it's very flexible. You don't only have to use FM index here, but because we were actually interested in lower memory and lower fast query, that's why we decided to try FM index, and it also allowed us to like compare against like how better does computing Simplitigs or SPS spectrum preserving string set is as compared to doing FM index on top of Unitigs, right? So that's why we chose the FM index route for building the membership query index. And because the size of the structure depends on the length of the string, making it a spectrum preserving string set improved the performance of the size of the index.
So spectrum preserving string sets are a compressed representation of the original k-mer set, but it would also be possible to further compress it, right, Amatur? And this is something you looked into?
Right. So there exists special purpose compressor tools which work which are designed specially for FASTA files, and they have much higher compression ratio for FASTQ files. So we applied one of those compressor tool on the output of Simplitigs or spectrum preserving string set to store the set of k-mers in more space efficient way. If we use spectrum preserving string sets, we improve the space usage by 10 to 28% compared to just storing the Unitigs with compression.
Karel, at the beginning you mentioned many areas in which k-mers are used nowadays, right? And how do you envision applying Simplitigs in those areas?
We envision Simplitigs to be used as a generic method for serialization of the k-mer sets on one hand, and as a very useful method for representing de Bruijn graphs for all the applications which are mentioned at the beginning, with the exception of genome assembly. So, for instance, in metagenomic classification, in, for instance, genotyping, and other applications. One of the very important features of the Simplitic representation is, and I would like really to emphasize this, is that it really follows the Unix philosophy of small tools which can be connected and combined together. Simplitigs are very easily computable out of a k-mer set on one hand, and on the other hand, since the output is just a FASTA file, it's very easy to combine them with different full-text indexes, with standard Unix tools, for instance, like grep or Perl and other tools. So we believe that it's very well designed, basically isolated block which can fit within standard bioinformatic pipelines.
You both did an experimental evaluation of your tools. You took slightly different approaches to it because your interests were perhaps a little bit different. Can you talk about like which approach each of you took and sort of compare them?
So in our case, we were very interested in the scaling. So the first question which we were asking was, how do both of the representations, I mean the Simplitic representation on one hand and the traditional Unitic representation on the other hand, how do they perform when we are increasing the k, when we are changing the k-mer size? The reason why we were interested in this is that different practical applications call for different k-mer sizes. So for instance, for people who work with nanopore sequencing, which has a huge amount of errors, people typically use small k. Whereas when people work with data which come from Illumina sequencing, people can afford larger Ks. So there was this natural question, like whether the representations behave always the same or whether they are different. And the results surprised us a lot. At the beginning, when we started with like very small Ks, what was happening basically was that the number of k-mers was growing exponentially, which is very natural, because when you have k-mers of length, for instance, 2, you can be sure that all the k-mers will be in the data set. Also for the length 3, and so on. So for a small case, basically the De Bruijn graphs are, let's say, saturated, or they are almost complete, which means that the unitic representation will be much worse than the Simplitig, because basically every k-mer will be branching. What we observed is that for every species, or for every model organism which we were working with, there is a certain point where the difference is maximal, and this point is typically logarithm of fourth of the length of the genome. And then the difference in the performance between Simplitigs and Unitigs goes down, and for much longer k-mers, they perform similarly well. So, for instance, in bacteria, this happens already for like k equal to 18. So Simplitigs and Unitigs will perform comparably well for k-mer of length 18, whereas, for instance, for humans, even with k-mers of length 31, there still is a big difference between Simplitigs and Unitigs. The other question which we were asking is, how well does the representation scale when we consider pangenomes? In our case, we were interested in bacterial pangenomes, which means we were basically taking...
Sorry, can you define what a pangenome is?
So there are many different ways how a pangenome can be defined, and it slightly differs between biology and bioinformatics, and in this case we are interested in so-called computational pangenomes. What we mean by that? By a computational pangenome, we typically mean a reference sequence plus all the within species variation. This leads us to the question, basically, or it's very largely motivated by the question, how to efficiently, or in general, how to represent all the variability which can appear within a species. For instance, bacteria are very highly variable, so they are always challenging as for the capturing all the variability. From the practical point of view, for instance, if you use a reference, for instance, for E. coli, which is too far from your strain which you are sequencing, all the methods you are using will be probably failing, because the many genomes won't be represented. So we were asking the question, when we are adding more and more variation, which means more and more gene content and more SNPs, for instance, how well does the Simplitig and Unitig representation scale? And what's the relative improvement of Simplitigs over Unitigs? And how we measured this? We basically took data. We took assemblies first from studies where they studied systematically the populations of bacterial populations in certain geographical areas. We were looking at how do the Unitig and Simplitig representation compare when we include more and more of the individual strains of this bacterial species. And what we observe is the following. When there's just one genome included, we observed pretty high improvements in the number of sequences, but almost zero improvement or very low improvement in the cumulative sequence length. However, when we were increasing the number of genomes included, the improvement was better and better with more genomes included, up to approximately 1.5 in our specific experiment. So this basically means that more data we have, more variation we have within our pangenome, the better Simplitigs are.
Amatur, what about you? How did you approach experimental evaluation?
So for us, we kept the value of k fixed at a single value because our a goal was to compare against different uh, tools that does the same compression technique as us, or the member does the efficient membership query as our method. So that's why what we wanted to do, we wanted to keep a fixed value of k and compare uh, and use different data sets instead in order to show that our method is scales well. So we use like a human genome data set, and we use bacterial data sets, and I mean we took different type of data set to show that our method worked well across all of them. And in order to show that our method works for large data sets, we used a human genome data set where the number of k-mers was around 400 millions. So that's what we did, but we never changed the value of k to compare like Karel did. So what we did was we compared against the tools that use membership query, that performs membership query in low memory, and we compared their time and memory requirement against ours and showed that how ours perform better in all five different data sets that we use. And we also showed the compression ratio of our method compared against other k-mer compression tools.
So if you want to store a set of k-mers efficiently and then query it for membership, one way to do that would be to run an algorithm, one of the algorithms that are called k-mer counters. And Amatur, I think you did a comparison of your algorithm with k-mer counters. So maybe you could talk a little bit of what k-mer counters are and how your algorithm compared to that. What did you find?
Right. So k-mer counters will take a genomic data set, and from that it will count the all different k-mers, and like it will output all different k-mers and their accounts, and they store it in a specialized data structure so that you can retrieve the count in a very fast and efficient way. So this is very useful in all the bioinformatics analysis. You use the k-mer counter tools a lot, but and they're very widely studied. So that's why we were interested in comparing our output to the output size of the k-mer counter tools. So basically, because in their structure they have the k-mers as well as their counts, we also wanted to in order to give a fair comparison, we also had our compressed set of k-mers, and we also had the counts of those k-mers in a separate file, and we compressed all of them and did a comparison against the size of these two data structures, the data structures that the k-mer counter tools use and the data structure that our tools used. And we found that our tools have a like on order of magnitude better disk space usage. So the reason is that in k-mer counter tools you have to pay the price uh, in space because you need that fast retrieval capability, but we don't do that. Our goal is simply efficient compression.
What do you think are the future directions of this line of work? Are there any open questions around Simplitigs?
I mean, you can definitely improve the algorithms. I think both of our work are, I mean, our algorithms are very simple for now. And for example, they're both single threaded, but we believe like both of them can be definitely highly parallelizable, and that would like speed up the process a lot. And also like you can improve the memory footprint of the algorithms. um, So yeah, so these are definitely something that we would like to work on. And another thing, like for our project, we use the computation of unitics as a first step. So I mean, that's like a very um, computation intensive task. So maybe if we just avoid computing unitics and start with the Bruijn graphs, uh, I mean, I'm guessing that we could get um, the same thing done in a very less amount of time. So these are some of the open questions. Like it's these are basically the questions that how to improve the algorithms that we implemented. Um, and also, if we another interesting question that we can look into is it possible to compress the set of gamers even farther than what we have? Like, what if we think of something other than simply the extent spectrum preserving string sets and reduce the disk space? That's something that would be something interesting to work on.
We sort of talked a lot about the methods and the algorithms, right? But there is an interesting sort of human situation here. I think a lot of scientists really dread being in your situation, which is like being scooped. And I'm really curious how you reacted, sort of what went through your head when you realized that someone else was working on the same problem. And I think looking at the dates on the preprints, Amatur published her preprint like a couple of days or like 4 days earlier. So Karel, what was your reaction when you like? How did you find out about this other preprint, and what were your feelings?
So we found the other preprint on bioRxiv, and our first reaction was, wow, we have to get our preprint as soon as possible. But we were really impressed because both of us worked on basically the same thing, but from very different directions. And I very strongly believe that both of the papers are very complementary. We were really very impressed.
Did you contact anyone from the other paper, like Amatur or Paul Medvedev, about this prior to like publishing the preprint, or did you just publish it and then let them know? How did that happen?
Yes, we got our preprint out. So at that time, our preprint was very close to finishing, but it wasn't done yet. So we had to like very quickly finish it within like 2 days to get it out as soon as possible. And once it was out, we contacted also the other group, and we were talking about the presentations and stuff like that.
And so, Amatur, what was your reaction when Karel and his team published their preprint?
Yeah, so I think I got to know about it first when I think Karel sent an email to me and Paul Medvedev stating about their work, and that's when I first found out that it's almost a similar work. Yeah, so yeah, I was excited. Like it's exciting to see that someone else is also working in the same idea as you, but they came up with the same problem in a slightly different way. But it's the method is kind of similar, so that was interesting. And the way I think the way Karel approached us, that was something from which I learned that he was very polite about it. Like we have the same kind of ideas. I think Karel also met Paul and discussed about a similar idea of greedily assembling greedily assembling KMRs. So I'm not sure about the history behind it, but I think they did meet before. Uh, I mean, the other author of my paper and Karel, I think they had some discussions about the same topic before. I'm not sure about that. I mean, I think Karel can say better about that. So at first I thought, what if I mean, did I do something wrong? did I copy someone else's work or? But then I realized that no, it can happen that you can come up with the same idea in a different from two different groups, and it's not that uncommon.
Karel, anything you want to add?
I think I don't have much to add, and I think it would be better to include this part in the podcast, because of course there was different teams are always working on similar things in some sense. Because basically the whole field of bioinformatics is kind of synchronized, or is the speed is basically given by the development of sequencing technologies, and different people do interact mutually at conferences and at seminars and so on. So of course also different people arrive at same ideas at the same time. So sometimes it happens that the same things basically appear independently in two papers, and I personally find it always exciting because it's very rare that both of the papers would be very, very same. You can always see that the papers kind of reflect the personalities of the authors, and it reflects the way how they approach the problem and everything. So I personally like it when something is basically being co-published or appears at the same time from two different groups.
Do you envision from this point on collaborating on the algorithms, or are you going to continue maintaining your own implementations and sort of exploring your own ideas?
Yeah, there are definitely some arenas where we can collaborate. Like we can merge our ideas. For example, like I said, we started with compacted De Bruijn graph, whereas Karel's method started with the De Bruijn graph, and that seems like a better way to go in both in terms of uh, like efficiency in memory. So maybe we can look into that and collaborate to make both of our tools better.
I personally can imagine that in the future we would come with a joint tool, kind of like next generation tool, where we would combine our ideas and where we would provide much better engineered solutions. Because in both of the cases, in both of the papers, the programs which we provided are more like prototypes. So there is definitely a huge space for improvement and for, let's say, providing one master implementation that would be highly optimized for memory, for the speed, and for other things, and possibly parallelized.
Awesome. Then I guess we'll be waiting for your master implementation in the meantime. And awesome work, guys.
Thank you.
Amatur and Karel, thanks for coming on the podcast.
Thank you very much for having us.
Yeah, thank you.